Hi, my name is Anna Ager, and this is our project, Biosensing with Waveguides and Landcover with Corbin Adkins, Michelle Eno, Benito Esposti, Andrew Kim, Justin Land, Nicole Schmidt, and Felipe Solorzano. In this project, we tried to design biosensors that would effectively detect the presence of mosquito-borne diseases, as well as determine whether or not the effectiveness of these devices could correlate with the Landcover data we analyzed this summer.
So first, it's best to compare current biosensing methods. We'll discuss four methods specifically, the first being reverse transcription polymerase chain reaction. RT-PCR utilizes DNA and RNA polymerase to identify changes in gene expression in order to diagnose disease. It's highly sensitive and specific, but it's limited to a disease's acute stage. Virus isolation, where a virus is separated from a serum sample, offers direct pathogen detection, but it's relatively expensive, time-consuming, and laborious. Next, viral antigen capture is a method of virus capturing samples using a specific antibody and offers simple and rapid detection, but lacks versatility as it's only viable for dengue and introduces the risk of false positive detection. Lastly, serology involves the complex study of blood serum and is similar to viral antigen capture in terms of vulnerability to false positives. And next, I'll get into Flaviviridae, which are a family of single-stranded RNA envelope viruses that share conserved structural and genomic features. And specifically, we looked into four different flaviviruses, those being dengue, West Nile, yellow fever, and Zika. And this is important because in areas where multiple different flaviviruses coexist, cross-reactivity between them may occur, which is when different antigens appear similar in detection. This can make accurate diagnosis a challenging task. So overall, this highlights the need for the joint operation of waveguides and accompanying contrast methods.
Next, we'll be discussing some of the specific contrast methods, the first being fluorescence. So fluorophores bind to the target biomolecule, making them stand out from others in a sample by having them emit light of a specific wavelength. Pros of this method are that it has a lot of potential for use alongside waveguides and is generally more accurate than the other method. However, because it requires impact from light of a specific wavelength, it's typically less portable and more expensive. The other method, which is surface plasma resonance or SPR, works by having a polarized light being fired at a metal film with a solution containing biomolecules on one side. The resultant beam, which has a dark band in the middle, will shift depending on how many or how few biomolecules bind to the ligands along the metal film. Pros of this method are that it doesn't require light of a specific wavelength like fluorescence, making it generally cheaper and faster. However, because of its very specific applicative properties, it tends not to be as accurate.
Waveguides are relatively simple electromagnetic biosensing devices. Light enters one side of the waveguide and undergoes a phase change due to the waveguide's internal structure. When a waveguide is paired with a contrast method, such as fluorescence or SPR, specific diseases can be tested for. The light in the waveguide passes directly through the contrast method and a sample, and by determining the exact phase change of fluorescence, the presence of a disease can be confirmed or denied. Here's our design for the waveguide. The light enters from the open rectangular end of the waveguide body and undergoes the phase change. The light then goes through the connector and into the vial chamber, where the light shines through a glass vial containing the contrast method as well as a sample. It comes out the other end, and depending on the phase change of the fluorescence, depending on the contrast method being used, the presence of the disease can be confirmed or denied.
These are disadvantages present in most other types of chemical sensors. However, there are promising niche areas and successful applications in biosensing that are of growing interest. The low cost and design flexibility adds to the appeal of the design, and future developments include possibility connecting the sensors into an optical network that will lead to the direct coupling of sensing systems to optical systems.
The SEES mosquito data and an ESRI population density map were observed to determine the contrast method. The mosquito points were generally concentrated in coastal urban regions, suggesting a relationship between human and mosquito populations. However, it is important to consider these observations were dependent on the residence of the SEES interns and may underrepresent certain areas around the US.
The maps on the right show the two subsets of the data previously described. They show isolated Aedes and Culex mosquito observations, which are the genera that spread flaviviruses. Most of these were in suburban areas near the coast where there was a population density of around 3 to 10,000 people per kilometer squared. By isolating these subsets, we were able to determine where the virus detection would be most needed and what design lends itself to the data context. With very few exceptions, these sites were accessible by road or short walk on open land, so the fluorescence method will work well given that it will not need to be very portable to reach these areas.
Data used in our project can assist the CDC in making decisions based on where specific species of mosquitoes are prevalent. The CDC can then make decisions about which device will be most beneficial within a specific situation and location. Hospitals can use their data and data collected from the devices to have up-to-date information regarding diseases in their area. This would allow them to be better prepared for any outbreaks. Communities could use the fluorescence device to gain information to know if preventative measures are needed.
In conclusion, we determined that we could use fluorescence and SPR contrast methods coupled with a waveguide to detect the presence of flavivivae, and we believe that land cover data correlates with the potential effectiveness of the device, considering that fluorescence-based biosensors would be most effective for areas with dense mosquito and human populations.
Thank you for listening. Does anybody have any questions?